This cow is not AI. It has a mutation in a gene that gives it incredible musculature and remarkably low body fat. And get this, researchers are working on a compound to mimic this effect in humans. I could not avoid seeing the parallels between our world and the Marvel Cinematic Universe. Cutting-edge science has a way of turning fiction into fact, sometimes faster than our imaginations can keep up. And just like in superhero movies, a lot of this happens in secret, behind closed doors, in the domain of the elite first and publicized second. But all of this is really happening right now. And that begs the question, how long until we are all superhuman? We already have a third generation of Ozempic-style drugs, and this is going to be a trillion-dollar category. In fact, there are similar drugs that also promote muscle growth, even if you have not heard of them yet. So what is next? A world free of Alzheimer's disease? Free of cardiovascular disease? The future is closer than we think. In fact, we might already be there. What if we could rewrite biology itself? This is where the comic book world and the real world begin to collide. If your DNA carried a mutation that caused disease, you lived with it or you died from it. The technology is here now, and over time it will inevitably become more affordable. Here is where things get deeply personal for me. Imagine biology that resets itself over and over again, maintaining youth and function indefinitely. There is another concept that sounds even more absurd until you see the data. In this video, we are going to start with where we are right now with human biooptimization, then leap forward, tying new innovations to their most appropriate superhero. We will discuss where the science stands today, where it is going, the limitations that remain solvable, and by the end, my goal is to convince you that a superhuman species may be approaching sooner than you think. Part 1. The muscle and fat problem. Let us start with where we are right now, and do not be put off by how mundane our first example sounds. The so-called miracle weight loss drugs of our time, the GLP-1 receptor agonists, medications like Ozempic and Wegovy that you have probably heard of. Regardless of whether you are a fan or a critic, these drugs are metabolically impressive, but they are far from medical miracles. One of their most important drawbacks is that they cause not only fat loss but muscle loss too. A meaningful proportion, about 25 to 39% of total weight loss based on human randomized controlled trials, comes from lean muscle mass, which is obviously not ideal. So the next obvious step in human bioengineering, particularly for body composition, is a therapy that does something better. One that selectively reduces body fat while preserving or even increasing muscle mass. That might sound far-fetched, almost like science fiction, except the data suggests it is already here. A paper recently published in Nature Medicine in 2026 suggests we may be on the cusp of developing a real-world prototype for a super soldier serum. The inspiration for this treatment comes not from Marvel, but directly from nature, specifically from a rare mutation in the myostatin gene, sometimes called the Hercules gene, found in double-muscled cattle, bully whippets, and a handful of humans who, through genetics, develop extraordinary musculature and remarkably low body fat. I say this lovingly. These people and animals are genetic outliers, and frankly, I am a little jealous. Aren't you? Interestingly, early literature suggests these mutations may even confer advantages for health span and potentially longevity. That is perhaps less surprising once you consider that muscle itself secretes signaling molecules called myokines that can protect other organs, including the brain. So what we may be looking at here is a genuine metabolic gold mine, or a vibranium mine, if you will. The trial I mentioned, a phase 2 trial, tested a fully human monoclonal antibody called bimagrumab. I know, it sounds like a supervillain. As a quick aside, I went down a rabbit hole trying to figure out why bimagrumab sounds so villainous, because I could not get it out of my head. And there is actually a teaching point here. Drug names like this follow a structure. The suffix mab refers to a monoclonal antibody, a Y-shaped protein produced by the immune system or synthesized in a lab that identifies and locks onto a specific target in the body. The segment GRU indicates that the drug targets growth factor or muscle-related pathways, essentially GARU, equals muscle growth. The prefix BIMA is largely arbitrary, chosen mainly to make the name unique. But the combination of strong consonants, the M and B from MAB and the G and R from Giaru, makes the word sound heavy and powerful. So completely by accident, the mix of pharmaceutical naming rules and harsh phonetics lands this drug name somewhere in supervillain alias territory. Now that I have given our hero enough time to escape while I explained my evil plan unnecessarily, let us get back to the data. The goal of bimagrumab, which is not a supervillain, is to partially mimic the biological pathway seen in these genetically gifted individuals by targeting the signaling systems that normally limit muscle growth, essentially taking the brakes off muscle growth while suppressing fat generation. The results of the trial were striking. Participants with obesity did not just lose weight. Essentially, all of the weight they lost came from fat. Even more remarkable, participants simultaneously gained muscle mass and improved strength and grip strength without any increase in physical activity. Think about that. It is like getting leaner and stronger while sitting on the couch, maybe even watching a superhero movie. This is an innovation that moves the human body toward a more superhero-like physique. Leaner, stronger, and more metabolically resilient. This is where the comic book world and the real world begin to collide, and it is not science fiction. This treatment has already completed phase 2 trials. It is here now. To be fair, and to temper my excitement a little, the treatment is not without drawbacks. Some participants reported mild side effects like gastrointestinal upset and headaches. But the larger point remains. We are beginning to glimpse a future where the human body can be directly engineered toward greater strength, lower fat mass, and improved metabolic health. Part 2. Rewriting the code. But this is barely the beginning. What if we could rewrite biology itself? This is where things get truly extraordinary and truly superhero adjacent, because the next generation of bioengineering tools does not just nudge hormones, it reprograms cells entirely. Let us talk about gene editing. Every cell in your body contains DNA, roughly 3 billion base pairs of genetic instructions. For most of human history, those instructions were fixed at conception. You got what you got. If your DNA carried a mutation that caused disease, you lived with it or you died from it. That era is ending. CRISPR-Cas9 is a gene editing tool that won the Nobel Prize in 2020, and it gave scientists the ability to cut DNA at precise locations, like molecular scissors. You cut through a strand of DNA at a specific site very precisely. The cell then repairs the cut, and during that repair process, scientists can disable a gene or insert new DNA. This makes me think of Spider-Man, maybe the best superhero analogy of all, whose powers come from being bitten by a genetically engineered spider, as if something like CRISPR merged the spider's DNA with Peter Parker's own, creating your friendly neighborhood Spider-Man. As a quick aside, I once had the pleasure of meeting one of the Nobel laureates who actually invented this technology, Jennifer Doudna, and I bring this up because she was as humble as she was brilliant. Scientists of that stature often carry a fair amount of ego, but she did not, and I wanted to acknowledge that. Sadly, she refused to turn me into Spider-Man. Bummer. Now let me show you why this matters with a real-world example. In December 2023, the FDA approved the first CRISPR-based gene therapy for sickle cell disease. If you are not familiar, sickle cell disease is caused by a mutation in the gene for hemoglobin. One wrong nucleotide, and that tiny error causes red blood cells to deform into rigid crescent shapes that clog blood vessels. This causes excruciating pain crises and dramatically shortens lifespan. For decades, the only cure was a bone marrow transplant, a brutal procedure requiring a matched donor that most patients never find, and even when they do, immune rejection remains a risk. Today, doctors can extract a patient's own stem cells, edit them with CRISPR, correct their DNA, and reinfuse the corrected cells. The patient's body then produces normal hemoglobin. Early results have been impressive. In clinical trials, 97% of sickle cell patients treated with this therapy were free of severe pain crises for at least 12 months post-treatment. Let me repeat that and really sit with it. A genetic disease that has tortured millions of people for centuries, corrected with a single genetic edit. What is next? Cystic fibrosis? Huntington's disease? Muscular dystrophy? You might wonder why every sickle cell patient has not already been treated. The honest answer is price. The therapy currently costs about $2.2 million. But consider this. The cost of sequencing a human genome has dropped from nearly $3 billion during the Human Genome Project in 2003 to between $200 and $600 by 2024, a reduction of more than 99%. The technology is here, and over time it will inevitably become more affordable. At least for sickle cell therapy, the obstacle now is not medical or biological. It is economic. Maybe we just need a couple of Bruce Waynes or Tony Starks to step in with some donations. If you will indulge a brief moral aside, I know some people consider this kind of intervention to be playing God, and therefore immoral. Personally, I believe that failing to act, when we have the technology in hand to end this kind of suffering, is the more immoral path. We are no longer in an era of simply managing genetic disease. We are entering an era of curing it. Now for a hiccup. CRISPR-Cas9 has limitations, in part because it relies on the cell's own repair process, which can be imprecise. CRISPR can sometimes cause off-target edits, inserting or deleting pieces of DNA it was not meant to touch. But there is more good news on the way: prime editing. A newer, more refined technology, sometimes described as the search and replace of gene editing. Instead of cutting both strands of DNA and hoping the cell repairs itself correctly, prime editing rewrites the target sequence letter by letter without ever making a full break in the DNA. If CRISPR is scissors, prime editing is a word processor: find, delete, replace, save. The precision is remarkable. In laboratory settings, prime editing can correct single-letter mutations, the kind responsible for thousands of genetic diseases, with minimal unintended edits elsewhere in the genome. This could be especially valuable when bioengineering more sensitive organs, like the adult human brain. Here is where things get deeply personal for me. I carry 2 copies of the APOE4 risk gene for Alzheimer's disease, the strongest known genetic risk factor for the condition. My lifetime risk of Alzheimer's is 10 to 15 times that of the general population, which is honestly unsettling. But here is the tantalizing question. What if we could change that burdensome APOE4 variant into the neuroprotective APOE2 variant? It would be the ultimate story of the villain becoming the hero, inside the cells that matter most, in the brain. In short, this could mean the eventual extinction of Alzheimer's disease, and it is not idle speculation. Research groups today are actively exploring this exact possibility using prime editing in the human brain. The real challenge is delivery, getting the editing machinery into the right cells in the brain. We are not there yet in humans, and the delivery problem is formidable. But real progress is being made using viral vectors, nanoparticles, and other emerging technologies that may one day turn this corner of science fiction into science fact. For someone in my position, and maybe yours too, a healthy person carrying a genetic ticking clock, the idea of one day walking into a clinic and having my APOE status rewritten is hard to fully put into words. It would feel like my own personal origin story, as exciting to adult me as the idea of being bitten by a radioactive spider was when I was 12. I still fantasize about that a little. Don't judge me, but sincerely, it is a remarkable thought. Part 3. Resetting the cell. Now let us talk about something even more ambitious than editing genes. What if, instead of fixing individual mutations, we could reset an entire cell back to a younger, healthier state? This is the domain of cellular reprogramming, and the superhero analogy here is obvious. Wolverine. He has the power of regeneration, healing from wounds that would kill anyone else. His biology resets itself over and over again, maintaining youth and function indefinitely. We cannot do that yet, but the biological foundation for something like Wolverine-style regeneration was laid in 2006, when Japanese scientist Shinya Yamanaka made a discovery so profound it earned him the Nobel Prize just 6 years later. Yamanaka showed that by introducing a few specific proteins, now called Yamanaka factors, into adult cells, you could reprogram them back into stem cells, capable of becoming almost any cell type. Think about what that means. Every cell in your body carries the complete instruction manual for building a brand new you. Here's an easy way to picture it. Most of the cells in your body carry all the DNA needed to make all of you. So what makes a liver cell, a heart cell, and an eye cell? It comes down to what happens as each cell develops. Your entire genetic code is like a book, and as cells develop, certain pages get glued shut, while others stay easily accessible. Certain cells simply have better access to certain genes. What Yamanaka factors do is peel back those glued pages, unlocking the full potential of the genome, potentially turning a liver cell into whatever cell type you need. There is a catch though. Fully reprogramming an adult cell all the way back to a stem cell is not what you want for anti-aging purposes. Doing that inside a living adult body would essentially create embryonic-like cells in places they do not belong. A recipe for tumors, not longevity. So researchers discovered something cleverer. Applying Yamanaka factors for a limited time produces partial reprogramming. This resets some of the epigenetic marks of aging without erasing the cell's identity. In other words, you can make an old liver cell behave like a young liver cell, or an old heart cell behave like a young heart cell, while preserving its overall identity. In a series of groundbreaking preclinical studies, this kind of partial reprogramming has been shown to reverse age-related epigenetic changes, improve tissue function and metabolic resilience, and in animal models of progeria, a disease that accelerates aging, it has been shown to extend lifespan. Multiple biotech companies are now racing to translate this into human therapies. We are not talking about decades away, we are talking about clinical trials within the next 5 to 10 years. The Wolverine analogy is not perfect. We are not getting vibranium claws or regrowing entire limbs, but the underlying principle, resetting biological age at the cellular level, is conceptually similar, and it is closer than most people realize. Part 4. Sending the message. Gene editing rewrites the code. Yamanaka factors reset the cell to a youthful state, but there is a third category of bioengineering that simply delivers a message, and do not underestimate how powerful that can be. Let us start with microRNAs, tiny molecules that regulate gene expression. MicroRNAs do not change the DNA sequence itself. Instead, they act like dimmer switches, turning gene expression up or down, which at scale has a massive impact. Your body already uses microRNAs constantly, both inside cells and between organs, to send messages. For example, your fat cells release signals to your brain that modulate sensitivity to the hormones that drive weight gain or loss. Your liver even sends microRNAs to your brain that can influence mood. MicroRNAs are part of how our organs communicate and adapt. What is exciting now is that scientists are learning to intercept, engineer, and deliver specific microRNAs as medical therapies. This is already being explored in several contexts. Cardiac repair, where microRNA-loaded vesicles are delivered to damaged heart tissue after a heart attack to promote regeneration rather than scarring. Neurodegeneration, using engineered vesicles that cross the blood-brain barrier to deliver neuroprotective microRNA signals. And metabolic reprogramming, targeting specific microRNAs to regulate insulin sensitivity, fat metabolism, or mitochondrial function. That is already impressive, but it gets even more interesting. There is another concept that sounds almost absurd until you see the data: mitochondrial transfusions. What if you could give old cells fresh mitochondria, like giving an old car a brand new engine? Here is the wild part. Mitochondrial exchange is already happening in your body right now, naturally. Supportive brain cells called glia grow literal nanotube tunnels that reach out and feed neurons fresh mitochondria. Fat cells shed mitochondria and send them to your heart to help protect it during a heart attack. This process is already built into our biology. The problem is that this natural renewal and repair process declines with age and is not quite powerful enough on its own to outpace the natural decline we call aging. But nothing stops us from synthesizing mitochondria in external bioreactors and transfusing them into people, repairing and enhancing organs the way Tony Stark might engineer an upgrade for Iron Man. Early trials are already exploring exactly this: growing mitochondria in bioreactors and transfusing them into humans. There is also a growing, more informal practice of people injecting themselves with mitochondria outside of clinical settings, though that is a different topic worth approaching with caution. I want to zoom out and get real with you for a moment. The superhero analogy running through this video was not meant to be cute, it was meant to be honest, because the gap between what comic book writers imagine and what we are actually capable of engineering in human biology is closing fast. Not through radioactive spiders or super soldier serum, but through real medical innovations that are rewriting and rewiring the biology that makes us human. The real question is no longer whether we will gain these abilities. It is how wisely we choose to use them, because with great power comes great responsibility. You know the rest. Like and subscribe. Health is wealth. Share this with someone who needs to hear it, and I will see you in the next one. Thanks for watching.